Our next talk and the last talk before the coffee break is Saket Navlakha from the Salk Institute, who is working on the connection between nature and algorithms in a different way in trying to really understand how we can learn and explain what learn from and explain what nature's doing in the language of algorithms. Cool. Thanks.
Okay, so thanks a lot for the invite, Carl, and thank you for being here. So really my main point today is to make the case that one future of algorithms in biology is to perhaps try and become the language for describing how biological systems process information and solve problems that are critical to their survival. And I think if you look around, it's not actually all that difficult to find instances where biological systems are doing what appear to be interesting computations. So whether it's an immune system efficiently identifying and eliminating a harmful pathogen from the body, whether it's the nose of a bloodhound efficiently tracking and homing in on a particular scent across large distances, whether it's a plant architecture that is growing very intricate geometric structures optimized for sunlight capture, or whether it's neural circuits in the brain that are still today a gold standard in terms of efficient learning and computation. These are just a few instances of biological systems that have evolved to solve very basic and fundamental computational problems that arise in nature. And I think this direction of looking towards nature for inspiration is certainly not new. So over the past several decades, there have been many instances of computer scientists turning towards biology and using some insights to develop methods for optimization. So for example, ant colony optimization, artificial immune systems, neural networks today. But I think really for algorithms to become a language of biology, it's really important for us to try to also reverse this arrow. To actually come up with ways that will convince biologists, or at least some biologists, that this direction is actually leading to insights that they might actually care about and that they can actually use to better understand how biological systems are working. And I think the occurrence of this feedback has been relatively less in terms of these kinds of methods, but one of the reasons that I think it's been that way is because of mechanism. Because we've observed these things in biology and we've developed some insights of how they might work, but really getting at the detailed molecular and cellular mechanisms that are driving these optimizations is, I think, a much better way to ground these kind of connections in actual biological reality in ways that we can then go in and try and predict the behavior of these systems. And I think trying to get at molecular and cellular mechanisms, this is a very exciting time to do this, mostly because of advancements in technology, just our ability to measure and probe and manipulate systems in ways that we've never been able to do before, and to really try and tie this thing together. And so really my main point today is instead of thinking of algorithms as what they've always been, the language of computing, the question is can they also, in the future or in the near future, also become a language for describing biological computing in nature. And I think what I think is one way to try to illustrate this point is by example. And so what I'm going to do today is give you an example of our recent work in trying to connect these two domains, and in particular studying a problem in neuroscience of how the fruit fly solves a very basic computational problem called similarity search or nearest neighbor search. Okay, and this is joint work with my colleagues in San Diego, Sanjay Dasgupta at UCSD, and Chuck Stevens, a neuroscientist at the Salk Institute. Okay, so let me start by describing what is this computational problem to begin with, how this is typically approached in computer science, and then I'll tell you about fruit fly olfaction and what this system has evolved to do. Okay, so similarity search is a problem that actually we solve all the time. Right? If I see somebody's face, I can look at this person and be like, oh, that person reminds me of Ben. Or if I hear a song, I can be like, oh, those guys sound very similar to this particular band that I like. Or if I smell an odor, I can be like, oh, this reminds me of a certain perfume that I've smelled before. Okay, and coming up with these connections is actually very important behaviorally because it allows us to generalize what we've learned in the past to some new experience that I'm experiencing now. So for example, maybe I should stop changing the radio station because there's a chance that I might actually like this song. Okay, but think about like what's actually happening for us to be able to do this. I've seen thousands of faces in my life, and then in real time I get a new query, and I have to somehow query this database of people or songs or odors that I've seen before and be like, okay, this is similar to these few instances. Right? And this is something that's quite seamless that we do all the time. And so how do we do this? And it turns out computer scientists in a very different direction have been studying this very basic problem for many decades now. And it comes up in very practical situations. So for example, if you go to Pandora, they have a huge database of songs that they've indexed. You say, hey, I like this song, and they suggest, okay, here are similar songs to the one that you're listening to and that you might like. Or YouTube, with video similarity search, news similarity search. This problem is a very popular thing in pretty much lots of applications. So let me start by telling you one basic approach of how this problem is solved in computer science, and then I'll tell you about what we've learned about how fruit flies do. Okay? Okay. So let's just set up the problem as an image similarity search problem. So again, let's say these are all the images that I've seen in the past, and then I get some query image, and my job now is to return to you the top k most similar looking images in my database. Okay, so how would you do this? So let's say that every image just has some representation. So you can think of it as some d-dimensional feature vector. These could be the raw pixel values of the image. They could be some SIFT or any type of higher level features that you encode. I don't really care. All I know is I have a point now in d-dimensional space, and I want to find similar images that lie nearby this point in d-dimensional space. Okay, so how would you find the top k most similar images? So on the one hand, this doesn't seem like such a hard problem, right? Clearly, you can just go through the database one at a time, compute the Euclidean distance or whatever distance function you want, and you can sort that list. Clearly, it will work, but this solution is just not scalable for large databases, right? You literally have to go through every one, so it works great, but only for small databases. And there's a whole other class of data structures, which I won't talk about, based on space partitioning methods like KD trees that will work for large N, but not for large D, not for large dimensionality. So maybe up to 10 or 20, but outside of that, you pretty much get reduced to linear search. And so this kind of tension between how do you solve similarity search with large databases and high dimensions has been kind of a very challenging problem in computer science. And it's motivated a different approach for this problem by relaxing this requirement that you solve this problem exactly, and you instead say, I'm going to try to solve it approximately. And the idea is that even if I return to you, k images that look reasonably close to your query, so users are going to be typically satisfied with this response, and even if they're not the exact most similar images. Okay, and so what do you get by relaxing this and saying, I just want an approximate answer? You can get a different class of algorithms to solve this problem, and one of the ones that is most popular is based on the idea of hashing. Okay, and this is not a conventional hash function. So if you have a conventional hash function and you have inputs, they are essentially by design randomly scattered over the range and over the bins in your hash space. And this is great if you're using this to store passwords because you don't want anything, any kind of structure here in the output space. But if you want to use hashing for similarity search, you would like your hash function to have a property that's called locality sensitivity, which means that two inputs that are very similar or look similar in input space are more likely to be assigned to similar or the same bin in output space. And if you can design a hash function that has this property, similarity search is actually very simple to solve. Right? All you do is you take your query, you find which bin it falls into, and then you just do a quick local search in that bin and return to the user however many images look similar. And again, this is not going to be perfect because there could be some scatter, but as long as it's good enough, you're happy with it. Okay? So formally, what exactly is a locality-sensitive hash function? It's a function that takes some d-dimensional vector, your input, and assigns it some point in m-dimensional space, and this hash function h is called locality sensitive. If you take any 2 points in this input space, the probability that they get assigned the same hash is equal to their similarity in input space. Okay, where similarity is just some function such as based on Euclidean distance or whatever similarity function you want. Okay, but basically the more similar you are in input space, the more likely you are to have the same hash. Okay, so how have people designed hash functions that have this property? Okay, and the main idea is to use what's called random projections. And to do these in a very low dimensional space. So if this is my d-dimensional input, what I essentially want to do is come up with a lower dimensional representation and doing this by multiplying my input by a random projection matrix. And so the idea is intuitively kind of like an unsupervised PCA, where if this is my input points, what I want to do is come up with some low dimensional representation. What's nice, and the reason that this works, is based on the magic of the Johnson-Lindenstrass lemma that says that 2 points that have some distance in this original space, that distance will be preserved to some factor in this lower dimensional space. And that's important, again, because if things are similar here, you want them to lie similar here so that you can most likely find them and return similar ideas. Okay, and what's important here is that this random projection is typically a dense Gaussian random projection matrix, which means that every element in your random projection matrix is just drawn uniformly from a normal 0-1 distribution. And again, the reason why you get this theoretical guarantee is based on the magic of Johnson-Lindenstrass, which I won't talk about, but that's the way it works. Okay, and so just to formalize this, if I want to come up with a low dimensional representation, all I do is I take my input x of I multiply it by this IID Gaussian random variable. You have to do some quantization to turn all this stuff into integers, but then that's basically all you do, and you repeat it 3 times, and now I have my hash for that input. Okay, and again, then how do you solve the problem? You take your image, you find what bucket it falls into, you find what else is in that bucket, and if I just want the top 3 guys, I'll just sort these guys in return. Okay, so there's many extensions and kind of bells and whistles on top of this core technique that you can read about, but this is what the basic idea is that has been used. Okay, so let me now transition into fruit flies, and I want to tell you 3 things. One is, why do flies even need to solve this problem? I want to tell you about what we've learned about what is the algorithm that is driving their ability to solve this problem, and then we'll do a little bake-off at the end comparing these 2 approaches. Okay, so let me tell you about fruit fly olfaction. So fruit flies, what is the goal or a goal of the olfactory system is to take as input an odor from the environment that it smells, and to assign this odor what typically is called a tag, or we can call it a hash, or literally just some representation. And this representation corresponds to some subset of neurons that are going to fire whenever that odor is experienced. Okay? And what we reasoned was that however this process going from the left to the right is, it seems to the fly's advantage that this representation be locality sensitive. And why is that important? Because let's say I'm a fly and I've learned to approach odor A and avoid odor B, then I'm never going to quite experience exactly odor A again, just because of noise and fluctuations and mixtures and all kinds of other issues. So however I process this odor, I would like to assign A prime a similar tag to A, even though I've never exactly experienced A prime before. So this is conjecture. This is our hypothesis, that however you do this, it seems that this function should be locality sensitive. Okay, and so we started reading into how does actually a fly in the brain go from an input odor to generate this neural tag. And it's a 3-step procedure that I'm going to describe to you in detail next. Okay, so here's the first step. So the first step, as an input, so a fly takes as input an odor, and it's originally represented by 50 odorant receptor neuron types in the antenna of the fly. So, and each one of these types fires at some firing rate. Okay, so you can essentially think of an odor as just a point in 50-dimensional space, corresponding to the firing rates of these 50 odorant receptor neuron types. Okay, and so the first step is that these guys project to, in a kind of one-to-one manner, to 50 glomeruli, or projection neurons in the glomeruli. And the result of this is kind of a mean normalization step. So if you plot the distribution of firing rates of these 50 receptor types, you get an exponential distribution, but the mean of that exponential depends on the concentration of the odor. Okay, so the higher the concentration, the higher the mean of the exponential. As a result of this projection and other inhibitory feedback that I'm not showing here, what you get is kind of a mean centering. So now every odor is represented by an exponential distribution that has roughly the same mean. Okay, why is it doing this? And so one of the ideas is that the fly does not want to distinguish between two different odors versus the same odor with two different concentrations. And so by doing this mean centering step, you're kind of fairly, you're kind of normalizing your data so that you don't make this potential mistake. And it does keep the concentration information around in a different part of the circuit that I'm not going to talk about. But this is actually, I mean, it was kind of interesting to see that this is often what we do with our data, right? We first normalize it, and then, downstream we go from there. So this actual task is called divisive normalization, and Rachel Wilson's group at Harvard has worked out in detail how this works. Again, I'm not going to talk about it, but this is the first step. Okay, so again, now I have my odor in this normalized 50-dimensional, as a point in 50-dimensional space. And what I showed before is that typically what we do then is reduce the dimension and then look and try to find similar things in this reduced dimensionality space. What the fly does is actually the exact opposite. Okay, so it takes this point in 50-dimensional space and blows it out into a 2,000 dimensional space. So it goes from 50 to 2,000, 40x expansion in the dimensionality. And amazingly, the connectivity matrix between these two is a random projection matrix. But it's not a dense Gaussian random projection matrix. It's a much more efficient random projection matrix. So it's approximately sparse and approximately binary. So what does that mean? So these 2,000 cells here are called Kenyan cells. And each one of these Kenyan cells basically samples from approximately 6 random glomeruli and sums up their firing rates. And that defines its firing rate. Okay, and there's been a lot of work on actually quantifying how do you know exactly this is random. I'm happy to talk about this later, but what you essentially get here now is your odor in 2,000 dimensional space. Okay, and now the last step is a kind of sparsification step. So each one of these 2,000 Kenyan cells provides feed-forward excitation to an inhibitory neuron called APL. And this inhibitory neuron provides feedback inhibition to each one of these Kenyan cells. And as a result of this feedback loop, about 95% of these cells shut off. They're kind of, you could think of it basically as a threshold, and everybody firing above the threshold stays on, everybody else gets shut off. And so what you're left now with is a very sparse point in a very high dimensional space, and this is kind of your hash or your tag or your kind of representation for this odor. Okay, so center the mean, blow things out into a high dimension, and then sparsify the heck out of it and get this point. Okay, so the first intriguing thing is, okay, this is a very unconventional type of random projection. Usually the analysis of Johnson-Lindenstrass has been dense Gaussian matrices. So the first thing that we did is, is this type of random projection also distance preserving? Because that would suggest that this could be useful for doing similarity searches. And what we showed was that if you take two inputs, x and x prime, and you get projected out into this way, into this m-dimensional space, you get the property that their expected distance after hashing is equal to, or approximately equal to, their original distance times some constant, where m here is the size of the expansion, and p is the 6/50 of how many ones there are in your random projection matrix. But this is just a result about expected distances. The reality could be different depending on your variance, if your variance is high. But we showed if you do order D random projections, then your variance is actually very concentrated around its expected value. And so what you get is this nice, the standard Johnson-Lindenstrass bound where the expected variance is 1 plus or minus epsilon the original times the original variance. Okay, so what this showed is that this type of random projection is a candidate for doing this type of similarity search. And so the way that I'm kind of thinking about is, imagine you have 1,000 people in a room, and I can stuff them all into this room and try to segment out and organize them by hobby. But then imagine I take these same 1,000 people and blow them out into a football field. It'll be much easier for you to kind of segment out and find structure in this data by taking advantage of this high dimensionality. And so what we want to do is kind of come up with a way to fairly compare these two approaches by trying to equate their computational complexity. And so the way we did that is by saying, okay, how many mathematical operations are literally used to compute this hash? So each one of these random projections requires 2 times d operations. So you have to do d multiplications plus d additions. And you're going to do that some number k times. And so what we did is that we equated the similar thing for the fly by saying, okay, the random projection is much more sparse. So instead of doing 2D operations, I just need to sum up approximately 10% or 6 out of 50 of the original indices. That means that I can afford 20 times more random projections to roughly get the same complexity. And of course, there's an additional step in the fly that you have to do this winner take all, but that's relatively cheap to do. Okay, and so we compared, in the final hash, you get the same number of active indices, but one is much sparser than the other. Okay, and the nice thing about similarity search or nearest neighbor search is there's lots of benchmarks that people typically use to evaluate these algorithms. So I'm just going to show you 2 examples. We've done many others. But the idea is that you take a query image, you do a brute force search through all your inputs, and you find the top 2% of the true nearest neighbors. And then you do your hashing procedure, and you find the top 2% of predicted neighbors, and you just compute their overlap of how close did I get. And then you can vary how many non-zeros there are in your hash, what is your hash length. Okay, and what we found, so these are just 2 examples. So the x-axis here is that hash length ranging from 2 to 32. The y-axis here is that overlap between predicted and actual nearest neighbors, and these are the 2 data sets. And what we see is that pretty much across the board, the fly does at least as good, if not better, than this standard LSH procedure, and it really wins the most in this low hash length regime where that additional dimensionality really gives a lot of benefits. So again, there's a lot of other evaluations that we're doing here, other data sets, larger data sets, other locality sensitive hashing variants, but we're still, we're seeing a lot of evidence that this kind of strategy is useful for solving this kind of similarity search problem. I have like 5 more minutes. Okay, so I want to tell you a few things about what we're thinking about next. Because the fruit fly is an amazing model system in ways that we can really probe the anatomy and the electrophysiology of these circuits. And so I'll tell you 2 examples of things we're thinking about. And so one is actually a learning step involved in hashing, which is how do flies or mice or even humans learn to discriminate between odors. So the example I like to give is, for example, with red wine. So I don't really drink much of red wine. So if you give me an odor of a Pinot Noir and a Zinfandel, basically their hashes or their tags are going to be exactly overlapping because I can't tell them apart at all. But with enough learning and through some procedure of reinforcement learning, what I can eventually get to is a step where these 2 hashes are separated out enough so that I can start to apply one behavior such as drink Zinfandel and avoid Pinot Noir. Okay, but think about what's happening there. So what's essentially happening is I'm getting hash collisions. Right? These 2 things are colliding, and how do we typically handle hash collisions? We usually double the index size, or we chain things together and then just do a search along the chain. What the brain seems to be doing is actually changing the hash function itself, which is actually a very different strategy because typically if we change the hash function, we need to rehash everything because everything could have changed. But by taking advantage of this kind of high dimensional sparsity, what we think is happening is that the hash function is just changing very locally around places of collision while keeping other things more or less as separated or using the same encoding for other regions of the space. And we think this is kind of a neat strategy for doing adaptive or online hashing where you're getting feedback over time, telling you how good your hash function is performing, and then as a result, your function has to adapt. So I think it's an intriguing problem to think about, both in terms of the basis of learning to discriminate and how we do that in the brain, but also in terms of this type of new type of locality sensitive hashing where you have some evolving notion of a distance function or a distance metric over time. So that's one problem we're thinking about. The other thing that we're really excited about is this problem of novelty detection. So there's really nice experiments done where you take flies and you expose them to a whole bunch of different odors that they're used to, and then you pour dust on the fly, and the fly starts to groom itself to get rid of this dust, and while it's doing that, you expose it to a novel odor. And when it's exposed to the novel odor, it immediately stops grooming itself and it'll start looking around. Because what's happened is there's something new, there's something latent in the environment that it changes its attention, and it realizes that maybe I should pay attention because this could be a good time to learn something new that's happened. And so flies are able to perform this novelty detection for odors. And so what we're asking is how do flies do this? Because again, the problem is it's experienced all these odors, it has a new one, and it needs to come up with some signal that says, yes, this is new, I've seen it before, or no, it's not. And again, we're starting from computer science, we're thinking, how would we solve this problem computationally? And one of the basis of this is answering the question, does X belong to S? Right? So here X is some query odor that you've seen or that you're querying. S is some list of previous odors. And you want to come up with some very efficient answer to just saying, have I seen this thing before? And one of the most popular methods, or space-efficient methods for doing this, is using, I won't talk about it, but is using something called a Bloom filter, which allows you to answer this approximately just using m bits of information, regardless of what are the actual content of these elements in S. And what we're finding is that, again, amazingly to me, that flies have also evolved variants of Bloom filters to answer this question of have I experienced this odor before. Instead of using m bits, it's using the synaptic weights of m synapses. And there's also some differences. So Bloom filter will give you a binary answer, yes or no, have I seen this before? The fly Bloom filter is a much more continuous answer. And instead of using random hash functions, if you're aware, it uses what are called distance or locality sensitive hash functions. And what this gets you is the analog of does X belong to S to the fly is how close is this new odor to some item that I've previously seen. And these two properties give that to you. There's actually a third property that we're also very excited about, is that this Bloom filter is also time sensitive, which means that if I haven't seen an odor for a long enough time, it's going to be considered as novel as if I've never seen it before. And this is important because in this regime, if I keep inserting things into this Bloom filter, eventually fill up and be totally useless. But here you just have some natural forgetting that happens. And so the analog question here is not only have I seen it, but how long ago was X or something like X observed in the past. And so we're kind of untangling the circuit that's actually doing this computation now. And then we're very excited about it. So just to conclude, in tying it back to the theme here, is that the question is, can algorithms be a language for describing how biological systems compute? And I think this is a direction that needs to be spearheaded definitely by computer scientists, but also, of course, in collaboration with neuroscientists or other biologists. And I think there's two main advantages to this type of direction. One is to computer science is that we can learn potentially new tricks to solve very basic problems that come up for survival. And I think to biology, it could be an attractive direction because what we could extract from this is quantitative principles that can succinctly describe and maybe even be used to predict the behavior of these systems and raise hypotheses and do experiments. So we're very excited about this. And again, thanks for having us. Thanks to Sanjoy and Chuck for working with me on this, and a plug as well. So thank you.
Questions?
So this is beautiful. Is the fly strategy evolutionarily conserved?
Oh yeah. So thank
Do
you.
the dogs do the same?
Yeah, so my one extra slide is exactly this. So I talked about fly olfaction, where you have 50 glomeruli. You have this sparse binary random projection to 2,000 Kenyan cells, and then you do this inhibition. So if you look at mouse olfaction, it follows a very similar recipe, just scaled up. So instead of 50, you have 1,000. Instead of 2,000 dimensional space, you scale up to 100,000. And not only that, but David Marr many years ago hypothesized that this motif is also something that you see in the cerebellum that's involved in motor control, and in the hippocampus involved in memory storage. And if you sum up the number of neurons in this column here, you get roughly 50 to 60% of the total neurons in the brain. So this basic idea of learning, discrimination, and identification of stimuli, we think could be a very basic computation that many other brain regions are using. So thank you. Yeah.
So you mentioned before that basically this hash function is adapting over time. And then you said later that basically you will forget something that you've seen too long before. Are these two things linked in that if you've seen something too long before, maybe the hash function has changed significantly enough that it doesn't hash the same thing anymore?
Yeah,
it's a different process?
Yeah, that's a good question. So I think there's definitely some mixing between these two, because if, let's say today I can learn to discriminate between these two, but I don't do it again for 10 years, then clearly it's probably going to take me longer to relearn that. Maybe it'll be simpler to relearn, but it won't be as natural as it might be today. So there's definitely some decaying process that's happening in that process as well. But we don't have much data about how those two forces are actually interacting, just because it's harder to do those long-term experiments over time. But it's a cool question. Thanks. Yeah.
So what you're saying about the dimensionality expansion, that's what people use in support vector machines.
Yeah.
It's been around for a while
Yeah.
in the computer science community. So may it be associated with the highly nonlinear responses that you have from the sensors, odor sensors? And the second thing is, how do you deal with the fact that there's so many combinations of odors in the air at the same time? You need to do some form of group testing or basically compressive sensing to find out heavy hitters and get rid of these noisy odors.
Yes. So I'll start with the second part. So the noise, I think one advantage of this winner-take-all that happens is that you do get rid of some of the noise so that only the very dominantly firing neurons stay active. And there was this really nice experiment where, let me just go back for a sec, what they did was they actually inhibited this inhibitory neuron optogenetically. And so what that means is that instead of getting a sparse representation for the odor, they had a dense representation for the odor by basically knocking this guy out. And what they found was that flies, if two odors were very dissimilar, it was still fine. It was still able to discriminate it. But two things that were more similar, it was not able to discriminate it as well. And that's exactly coming out because that this sparsity is really highlighting the things that are dominantly different between the two, and then that helps it. And the fact that they showed it experimentally, I thought was beautiful. You're right.
There is a method in compressive sensing that does threshold.
Yeah, the iterative hard threshold thing. Yeah, so we're trying to find connections between these things, and I think there's, even at the receptor level, if you think of what odor space is, you have, potentially millions of possible mixtures that you can form out of basic odors, but all of that is being compressed into 50 odor receptor neuron types. So there's some work that we're not doing, but other labs are doing, is understanding what is that relationship between chemical odor space and this representation, and is it a type of compressed sensing that's happening? So I think it's a cool problem.
Any other questions? Yeah, that's nice. Again.